welcome back. And here's that information about that Tufts University veterinary medicine program visit. They're interested in talking to students who are interested in animal sciences, and they're going to come up here in, I guess, a week or two, a couple of weeks like that. And so there's just that information that I showed before. And notice there's some signs up and around the science building with that scan code on it. And so we did our first exam, and we'll talk about that, and we'll follow up on that. And so what you should do this week is when I post today's lecture, I'll post the answer key video, and so you should go over that. I also took a picture of a couple of answers that specific students gave to show, just they're anonymous, but just to show you, demonstrate some solid answers like that. I do that in the video too, but I wanted to show you evidence of how students are answering questions. And we're going to have an assignment, and the assignment is to force you to follow up on the exam. And it says find 20 points you could have answered better. So maybe you didn't get it wrong, but you'll see in the answer key video, you could have had a better answer, because you might have gotten all the correct points, but there would have been an even better answer. And so that's not going to change your grade on the exam. It's just a new assignment, forcing us to follow up on that. This exam is not worth as much as the second, and that exam is not worth as much as the final. And so a big part of what we're doing is, this was unexpected, and most students did not do as well as they want to do in this course. And so that's built into this. We're trying as hard as we can to learn this material with assignments and quizzes and so forth. And if a student asks me, hey, how can I do better in this course? I talk about it in the answer key video, and the answer is focus on what's happening in class. All the points on the exam were from class, literally. You can expect some points from the book, but the big thing is to tackle the course by finding out what's going on in class. And so if something comes up once in class, you should be scrambling to write it down. Most of the stuff is represented in the book, but it's more disorganized. I'll say kind of more disorganized, I feel. And so that's the big focus. And then it's all about recall, And so then the next thing to mention is it's unusual. This is an unusual exam in that more than half the points came from our assignments and quizzes. Like just recreate that WINT assignment, recreate the muscle differentiation assignment. And so those are really important. And we had answer key videos, they need to be dealt with like that, right? So we'll follow up on it. I'm happy to talk with you about it. And I've taught this course many times, and it's always the same. No matter how much I try to set it up, students are stunned by the first exam. They're just like, oh wow, because we have to be able to explain the stuff in your words like that. Okay, so we have that assignment, and then a little shift from the syllabus. We're covering and I mentioned this last week. We're covering the chapter on plants next. So we'll jump into that today. And I'll post a video that I made that's got an assignment that's all described in the video about understanding flowering in plants. And so we will get to that today and talk about that. And that assignment will be due in a week from now. So that's what we're flowing forward on. This is not our course. This is an intro bio course, and I just want to show you what I do every time I give an exam. And what I do is I show an anonymous distribution of the scores. So on this intro bio course, there were lots of students in this course. There was a total of 80 points on the exam, and the asterisks are individual students, anonymous students. So here there are two students that got 57 out of 80, and I gave them a C+. So I don't grade by a 90, 80, 70, 60 cutoff. I don't do anything like curving. I was a student where I went to college. My friends and I talked about grades all the time, and we hated lots of grading strategies. What I do is I've taught our course a bunch of times. I just graded it, and I look at that distribution, and I say, yeah, well, those students get an A. Maybe nobody got an A. Maybe I'll say that. Nobody really got an A, I feel. Like that. Best student got an A minus. And I'm not looking at the 90, 80, 70, 60. It usually comes close to that, you know, but a little gentler than that, and it's not a curve, it's just me looking, and what I'm trying to do is I'm trying to give everybody the best grade I can. And I want to show people this because somebody will say, well, why did I get a C plus? And the reason is because you would have had to get those points to get a B, you know, like that. And if you'd gotten fewer points, you would have gotten a D. And so I think it's important. You're not competing with each other. Don't worry about that. It's not about that. You're not competing with me. I'm trying to give you as good a grade as I can, and I'm, you know, have to judge, you know, how did you do on the exam. This course is difficult because they're chunks of points, right? One question is worth 10 points, and you could just be like, yeah, didn't get that, right? None of the big questions are like, you know, a footnote from the book, right? We talked about it, or we had an assignment on, you know, they're big chunky things, but the day of the exam, you're like, oop, I got nothing, or you got off track, you know, you get off track and you lose a bunch of points. So points here, points there, and those add up. And so, you know, I don't focus on, you know, trying to write an exam where somebody could get 100 on it like that, you know? And so, you know, I'll post our course distribution on our Brightspace when I post the answer key video. So you'll be able to see how you did. And what it's like is you go on your exam and there'll be a circled score and you got like 67 out of 100, okay? And you just take that 67 and you'll find your asterisk on that distribution and it'll tell you what letter grade you got. The exams are worth most of the points in our course, like that. You know, we have a lot of quizzes and assignments there. In total, they're worth a good amount, but you know, the exams are still the big thing. And so, you know, if you're getting your assignments done, you know, and getting those, you know, completed, you'll get that chunk of points, and then, you know, these exams. And so you should be aware and comfortable about where you stand. What's your letter grade for this course? You know, you're getting a bunch of feedback, and this is more feedback. And we can talk about it. And, you know, as we get closer to the final, you'll say, oh, what do I have to get in the final to get this grade? That's actually described in the syllabus, but you and I can talk about it too. Yep, okay? So that's that. And we want to follow up on it. You know, we're not going to walk away from this exam. We're going to follow up on it, and that's one thing. I also want to just point this out. I said there could be 20-point questions, and instead of writing just one flat-out, open-ended 20-point question like this, you know, where I just say, hey, tell me everything you know about the vertebrate phylotypic stage. What I did is I decided to break them down. So 12 points and 14 points, that's still a lot of points in the question. But, you know, if anybody was surprised by that question, well, that's shocking because we talked about it so much and emphasized so much. This could be a big, chunky question. And it was, but instead of just doing it open-ended, I said, tell me this, tell me that, tell me that. So each of those are 2 points. Same thing down here, that's that frog embryo molecular mechanism handout. I could have just said, go for it. And I will on exams, but I just decided on this exam, you know, to try to set it up, to kind of break it down a little bit, to guide you a little bit. So that's what happened in that. So there wasn't any just great big open-ended essay assignment. There were plenty of long answer questions, you know. Like the Wint drawing assignment, I say draw it out, label it out, and then also explain it, you know, like that. So that, you know, students produced a lot, okay? So that's that. And, you know, we'll loop back on it. If you have questions after you go through everything or you're confused about the assignment, how to get this done, we'll meet, we can talk about it. You can bring those questions to class. We're all following up on the exam. I can make mistakes, you know, like that. And certainly when I make the answer key videos, I try to go through them kind of quickly, you know. So sometimes that explanation is not fully, you know, clear on, you know, what about your answer and what was wrong with your answer. So we want to follow up on that. The final exam covers everything, and we're building up like that. I just want to put a plug in for this course that's taught every other spring. It's going to be taught in the spring. Taught by Rachel Simons. Many of you have had Rachel. Rachel's a fantastic teacher, and the course is called Comparative Vertebrate Anatomy. So Rachel, if you had her, Rachel normally teaches courses on human anatomy and physiology. And this is not limited to humans, it's all vertebrates. So that course very much complements what we've been learning about in the first part of our course. Because that course, and I don't know that Rachel ever mentions the vertebrate phylotypic stage, I don't know, you know? But that course starts at the vertebrate phylotypic stage and describes the great diversity of vertebrate anatomy. So in vertebrates, every vertebrate has a heart, but there are 2-chambered hearts, 3-chambered hearts, and 4-chambered hearts in vertebrates, like that. There are kidneys in every vertebrate, but they have a very different story due to the diversity of vertebrate bodies and their evolutionary history. And it all fits together in an evolutionary vision, but it's very nice as a follow-up on our course because we just don't go there. For example, we're just not going to talk about these branchial arches, which produce huge differences in the vertebrate body in this area. So fish gills, right, versus human jaws and a middle ear, ears like this, and so forth. And that's what Rachel's course is all about. So that's a reason why we don't touch on some of these things here, is because they're touched on in Rachel's courses really well, and they complement each other. So I just want to plug that course. If you have questions about it, let me know. Today, what we need to do is we need to kind of follow up and extend some things that were part of that first section of our course, which was early vertebrate development. And these are some topics that were really covered in chapter 4. And remember, I said, let's just not read chapter 4. So you don't need to read chapter 4, but this is material that can be on our second exam and final exam, okay? And there are just these 3 kind of topics that the book deals with as well. And some of them the book deals with in chapter 1 and 3 also. And I just want to kind of bring them home to us more. So before we get to plants, we'll just talk about these. So the first one is this experiment that scientists can do on mammals. And what they can do is take a pre-implantation mammal of any species of mammal, and they can split the early embryo and make identical twins. And that happens naturally in nature. A scientist could do that in a lab as well. They could manipulate the pre-implantation embryo, split it, and make identical twins. Or split it again, make identical triplets or quadruplets. And that, again, happens in nature. It's just a phenomenon. And what it's saying is that all the cells in the early pre-implantation embryo are equivalent. Each one can make the whole organism. And a complete organism, a full-sized, mammal with all the organs and, just living a fine life. And those cells are given a special name. They're called embryonic stem cells. It's just a name to focus on the special quality of those. Those are only found in mammals. There's no such thing as an embryonic stem cell in any other organism on our planet, okay? And so what this experiment is, it's just a cute trick thing is, well, you could also take 2 pre-implantation embryos and just put them together. So take these 2 early balls of cells and just put them together and put that back in a surrogate mom, and there'll be one baby born. And they used a different fur color here just to depict that this early embryo came from a mom and dad that is a brown furred mouse. And this early embryo came from a white furred mouse. And when you see their babies, these babies are composed of cells from 2 different sets of parents. And they're just bombed together. And so this specific mouse will randomly have some brown fur and some white fur on its body. And it'll vary. And so here's an actual, I guess I didn't include that picture. I have it somewhere. I think I just got that one picture out of order because I have a bunch of pictures. So here's just a litter. And these mice were made individually in a dish. They thought, we're going to blow this one, put this one together. As you can see, here's one that's mostly brown fur. Here's one that's mostly white fur. And it's just a chance thing, what they contribute, like that. And that's because the fate map is not set up when we put them together. All the cells are equivalent. They can make anything. So it's a chance thing. And if you took their tissues out and looked, they have either the kidneys made from either the white furred parents or the brown furred parents, like that, yeah? And so those genomes, the brown furred parent genomes and the white furred parent genomes, they don't blend together in this animal. These are 2 different sets of cells. It's really 2 organisms in one. If we took one of these and raised it up and bred it, what we would find is that the individual mice will make sperm or egg only from the white furred parent or the brown furred parent. So this is an unusual generation. It just happens once, and they're called chimeric mice. Sometimes they're called mosaic mice. Chimera is from this ancient Greek word where you could have a horse with a dragon head. So that's a chimera, is a hybrid organism. And so these are chimeras. Just as a side note, every time I teach this, a student raises their hand and says, oh, this can happen naturally, very, very rarely in humans. So you'll see, you can find on the web, there'll be some person and they discover, usually as an adult, they're actually composed of two different humans put together. So that does happen, but I think currently on our planet there are only 6 or 8 people who have that situation. And what happened in their bodies, just to say, because some students spot this, is what happened in their bodies is that that mom and dad were going to make fraternal twins, and we haven't mentioned that, but the difference between identical twins and fraternal twins is identical twins are one sperm and one egg, and they start to develop pre-implantation and they split. So those are identical twins made from just one sperm and one egg. The identical twins have the same genomes. Fraternal twins, on the other hand, are the same as a brother and sister, as siblings. What happened there is, by chance, mom happened to ovulate 2 eggs in one cycle, and those 2 eggs are totally different. And then dad had 2 independent sperm, fertilized those 2 eggs, and it's just 2 siblings. Fraternal twins are just siblings. They could be brother and sister, whereas identical twins are always the same sex, right? So those are fraternal twins, and what happens super, super rarely, you're never gonna meet anybody this happened to, but super, super rarely in humans, you can have 2 pre-implantation fraternal twins, and they get stuck together and grow as one. And so the person is a hybrid, a mosaic, a chimera, okay? And they're 2, different sets of organisms. Those people would also just send on one set to their offspring, sperm and eggs. So back to the experiment, these mice will just send on one set. And we don't know which one. This white furred mouse in this generation might only be able to produce brown furred offspring. It's just a chance thing, but it's just one or the other, and it's 100% either brown furred or white furred. So we have this first generation and then the next generation, okay? And so scientists knew this, and it's just some kind of curiosity of the experimental lab, right? It's happening, the story's happening here, and the way the scientists do that is they do in vitro fertilization. So they gather sperm and eggs from the mice they want, they fertilize them in a dish, they let the pre-implantation embryo grow in a dish, they do whatever experiment they want, and then they implant it in the uterine wall. So same as medical in vitro fertilization is in people, just doing little kind of games to learn more about biology, okay? And so we have the idea of identical twins, we talked a little bit about that, and the idea that these are early pre-implantation cells, okay? So then the next thing is that these scientists won the Nobel Prize, I don't know, about 20 years ago, because they used just that quirky thing to do something. So it's a quirky, goofy thing, but they did a special experiment that became a very valuable tool. And the work includes 2 stories. And the first story is just this, that we could put 2 embryos together and make a mouse that's this hybrid mouse, okay? So what they did is this. They took an early pre-implantation mouse in a dish, and they added a cell that they were growing in a dish. So remember, we learned that you could grow embryonic stem cells in a dish. So they could add that to the embryo, and that would be included in the chimeric mouse. And so that they realized that they could take these embryonic cells in a dish, they could manipulate them. They could change their genes in a dish, and they would just have individual cells in a dish. But that first story is a way to take special cells in a dish and turn them into a mouse. Because, we'll just go back to this,